already told about the title, so I have to present my. Okay. Here are my co-workers, as I said before. All started with when Professor Mauro Ferrari, he was short time also president of ERC, but he was before director of the Texas Health Center and then Houston Methodist Research Institute, so he invited me there, and where I learned all about tumors. There I met Paolo De Cuzzi, with him I work now quite a lot. He's at IIT in Genoa, and his group is here. Is Valentina will show some results, and Tom Limor. Then I work mainly with Professor Wall in Munich, and his group, Johannes Kremheller and Barbara Wirtel. And I shall also deal a little bit with artificial neural network, and that is Daniela Borso. And regarding theoretical framework, we worked with Bill Gray and Casey Miller from North Carolina. So the outline, I will shortly introduce. I mean, we work within oncophysics. So I will tell you what is that, then I show you our model, the numerical solution, and validation, and then I will deal with drug delivery via nanoparticles and the use of artificial neural networks for several applications. Now, oncophysics, or physical sciences in oncology, was an initiative by the National Cancer Institute. It started in 2007, and the aim was to see if physics could help to understand cancer better. And we work, there are many areas, but we work mainly in transport oncophysics. And the paradigm there is that the physical properties of biological barriers control cell, particle, and molecular transport across tissues, and this transport and its deregulation play an overarching role in cancer physics. But there's also another aspect which I will deal with. That means probes, in particular nanoparticles, which are used to investigate the mass transport properties of tissues, can be used as directed vectors for the localized preferential release of therapeutics in the tumor. Just to tell how we see the tumor, I said this deregulation. For instance, tissue invasion is deregulation at the interface between the healthy and the tumor cells. Metastasis is deregulation in the cell transport. Then tumor-associated angiogenesis is deregulation in fluid and mass transport, and alteration of the signaling pathways that have to do with evasion of apoptosis, growth signal dependence, and growth inhibitory message is again a deregulation at molecular transport. But it's not only transport, also the forces are very important. For instance, I mean, there was a paper in 2012, and it stated the forces generated by tissues and acting on them influence the way tumors grow, develop, and metastasize. And that's important. These forces may even be as influential as the genetic and molecular changes at the focus of modern cancer research. So we work in this area. So I show you our model, which is a multiphase flow in the form porous medium. There was a paper again in 2012 about soft matter models developing tissues and tumors, and we apply or make use of the analogies between tissue and liquids. And in particular, cells and tissues can be treated as continuous media, and the rheology is described by macroscopic properties such as surface tension and viscosity. I will address that shortly. And we use as a solid part the extracellular matrix, which is the structural component of the cell microenvironment. Just if you look at the video, that is cell migrating in a 3D extracellular matrix, it looks like flow. I mean, so the model is quite justified. So I said one important part, which many models do not have, is the solid phase. which is the extracellular matrix. In healthy state, it's here. I mean, you have the blood vessel, lymph vessel, and then you have these fibers, which are sugars in some way, and collagen. And here again, hyaluronan is again a sugar, and then fibroblasts are cells of this stroma. However, the extracellular matrix exists obviously also in the tumor, but there it is disorganized. Compare the two. I mean, we had each component have many more components, and sorry, what happened here? Many more components, and so the model must be able to represent also this remodeling of the extracellular matrix. And the components of our model are 5 phases. We have the extracellular matrix, we have the neovasculature, we have the interstitial fluid, then we have a tumor phase, which is composed of 2 species, the living and necrotic tumor cells. Then we have the healthy cells. Then other species are nutrients, tumor angiogenic factor, and here's also drugs, which I forgot to add. How do we get the model? I don't want to show, I show just one slide with the equation. Okay. We start writing the relevant balance equation at microscopic level, including also thermodynamic statements, which are the standard irreversible thermodynamics. And then we scale everything up to the macroscopic scale using the thermodynamic constraint averaging theory, which was introduced by Gray and Miller, and come up with the balance equation at macroscopic level. And we get also a simplified entropy inequality from which we get the constitutive models. Just by upscaling here, you see again the 5 phases with the species. Through upscaling, we get the volume fraction, which are here, volume fraction of tumor, healthy cells, and interstitial liquid. The sum of them gives also the porosity. Then we have the volume fraction of the neovasculature and the volume fraction of the solid, and they have to sum to 1. Why it's stuck? Okay. With, as we said, we have also angiogenesis. So we deal with the new vasculature in a homogeneous version, while we resolve the pre-existing vasculature as embedded one-dimensional inclusions with blood flow and species transport. Obviously, we need suitable coupling constraints to couple blood flow and species transport in the pre-existing 1D vasculature and the 3D homogenized neovasculature, and we use a motor type formulation for pseudo numerics. And what is nice that the 2 meshes are independent. So the extracellular matrix gives to the existing vasculature vessel compression, which triggers the angiogenesis. From there sprouts new vessels to the tumor, and from there we get fluid, oxygen, and drugs, and so on. We have also to define the exchange terms, just to tell you what we all take into account. I mean, we start with the blood in the existing vessels. The chemical species are exchanged with the interstitial fluid, which brings them to the living tumor cells. The living tumor cells grow and remodel the extracellular matrix, but if the nutrients are not sufficient or the pressure is too high, they go in hypoxia and become necrotic. But before they become necrotic, they send out the tumor angiogenic factor to the blood, then new vessels sprout to the tumor. Then again, it starts all over again because the new vessels can bring again nutrient, and so the whole thing starts. And the necrotic cells are degraded, the membranes, by the macrophages and revert to fluid. So that is okay. And the host cells are in homeostasis. I told that the important part is the viscosity. We use the dynamic viscosity, some sort of stick-slip behavior. That means that the cells do not move if the gradient of the pressures is below a threshold value, and then they start flowing. In this way, we can model adhesion. And the surface tension, as I mentioned at the beginning, they come in through the pressure difference saturation relationship. I mean, for those coming from porous media would be the capillary pressure saturation relationship, but here we can't say that because capillary pressure is the blood pressure in the capillaries. Okay, so this comes from geomechanics. Here some improvement would be nice, but they work. We use these relations, and here we have the scaling factors where the surface tensions are in. For the mechanical behavior, we use either Neo-Hookean law, we use the effective stress principle, so we need the solid pressure, which is given here, and we use a total Lagrangian approach, or we have also a elastoviscoplastic one, Bergina type for remodeling. Now we have the equations. We get the weak formulation with the Galerkin's weighted residuum approach and discretize them with finite element in space and come up with this system of equation. You have 7, then there would be an 8th one for the drugs. I mean, the variables are nutrient, mass fraction, tumor angiogenic factor, mass fraction, then a neovasculature volume fraction, because that is a phase, then the pressure difference between tumor and healthy cells, pressure difference between healthy cells and liquid, interstitial liquid, then pressure of interstitial liquid and the solid. We solved that in time with a generalized midpoint rule and Newton-Raphson. Now I have to show you that it works. I mean, it is now implemented in this code BACI, which stands for Bavarian Advanced Computation Initiative. Okay. Just to tell you what we can do, I mean, we can account for growth, hypoxia, necrosis, lysis, then mass exchange between the components, then invasion and infiltration of the tumor cells in the healthy tissue, migration through an existing extracellular matrix, deposition and remodeling of extracellular matrix, different interfacial tensions, then max exchange between newly created and coopted blood vessel and transport of therapeutic agents and signaling molecules. So the model is quite versatile. I have to show you that it works. So we did quite a lot of validation. I just show you one here. This one is glioblastoma multiforme. They have been grown in these plates, and there are these wells where these multicellular spheroids grow, and then we can follow in time the radius of the tumor and the days. And I mean, this one is quite easy, but there are other ones. I showed this because that's needed for the next slides. I mean, we are quite able to follow this one. Now, because several tumor growth models do not have the solid phase, I show you, or want to convince you that it's very important that they have a solid phase, because I show the importance of porosity, stiffness, and spatial organization of this extracellular matrix in the development of whole malignant masses. If we take again a glioblastoma, and it has been grown in fibrous collagen type gel, which resembles the organized structure in the brain, so-called spheral structures, and also in compact hyaluronic acid gels, which are more homogeneous, which replicate the brain parenchyma. And we show that the behavior is completely different for the same cells. You see it here in the collagen, that is after 1 day, you have a single cell. After 7 days to migrate in that organized structure, they become elongated, while in the hyaluronic acid, they have the cell round. They stay round even after 7 days and are prone to clustering. And also here in collagen, you have this nice corona, you have infiltration, which here you have it immediately after 3 days, while here you have to start in the hyaluronic acid, you have to start 6 days, and the corona is much slower. Also, here you see the radius. If you see how the nucleus grows, it's increasing, while in the hyaluronic acid, it comes to a steady state. Okay, some sort of steady state. Now we try to model that. And you see, for the hyaluronic acid, it's quite easy. I mean, we're immediately able with the porosity of 0.8 and Young's modulus of 200 Pascal to simulate the behavior. It was quite satisfactory, but it was much more difficult for the growth in the collagen. If we started using a homogeneous medium, we are with different porosities, we are quite out. These are the experimental values. Then we started to introducing sectors to show this character, the fibers with different porosities. We did the first choice, which was again not good. The next one was slightly better. However, we remembered that at the beginning, I mean, these different shapes of the cells are only after 7 days. So at the beginning, the cells do not feel how is the extracellular matrix. They just see the local environment. So for the first few days, we kept a homogeneous structure, and then the fiber structure, and now we were quite able to follow this. And you show here what we get after, I don't know how many days, but this one is the tumor, heterogeneous. You don't see the corona because the density is too low for the graphics. Okay. So here is compares. I mean, that was in the growing medium, which I have shown at the beginning. Then that is in the hyaluronic acid, and this one is the collagen. So first conclusion that spheroids in suspension do not develop as rapidly and aggressively as spheroids embedded in more realistic environment. Then non-uniform porous matrices can be more efficiently support spreading and growth of tumor compared to uniform porosity matrices. And since we kept constant or same the adhesion and ACM modeling in both models, the major differences can be ascribed to mechanical cues, which is porosity, stiffness, and spatial organization. Usually people talk about stiffness, but we found out that much more important is porosity and the spatial organization. Okay, I show you some other now, a tumor in the host tissue. I mean, experiments that I've shown that this one is the glioblastoma, which eats its way into the astrocyte and the brain cells. And we investigated several. Here you get the tumor cuts, but let's look here with the spheroid away from the vessels. So the first one we did it avascular. So we didn't take into account the vessels, but we looked at the play, what happens with the interfacial tensions. Now, if we vary them and you get completely different behavior, you see here the radius in time, also the necrosis is different. And here with different ratios, the common case is this one is invasion. You see that the tumor growing, you have the living tumor cells, you have the necrotic tumor cells, and here it pushes out the healthy cells. Then with another combination, you get this infiltration, which I have seen in the experiment. I mean, the same radius, you have both living tumor cells and healthy cells. And that is the nice one. I mean, this we got now only numerically. That means that with certain ratios of the surface tensions, you get an invasion of the healthy cells in the tumor, which means that you could get less dense tumors, because one of the big problems is to bring the drug inside the tumor, which you shall see. So that would be a way, but that has still to be investigated, because that is only in silico for up to now. Now I show you another application. We take into account the existing vasculature. So a similar case. you see the tumor cells. These one are the isolines of the tumor angiogenic factors. And you see in days, now that is 12 days, 18 and 24, the tumor grows towards the vasculature, and on the other opposite side you have the necrotic part. You see the blood pressure and the existing vasculature and the neovasculature and the oxygen, and then the same on the tumor angiogenic factor in the interstitial fluid and the oxygen. Now these were all 2D or axisymmetric, but we can also do 3D. So that is growth of a tumor along a pre-existing blood vessel network. Here you see a few snapshots. The yellow part is the necrotic part, and here is the video. I hope it works. Yeah. You see the tumor growing, then inside now starts the necrosis. And after a certain time, you have this one. So the center is necrotic, and you have the outer is the viable rim. That's it. Now, one question: does our SME or hybrid model for angiogenesis work? So we did one of the largest validation exercises on reanimate. You can get the pictures, I mean all the data for, we got 3 tumors. I show it, sorry, no. Here there are 3 tumors that were grown in a mouse, and then all the data were there from imaging. So let's go back. So what we do, we solved it, either fully resolved the model with 20,000 nodes, and it's quite big one. From this one we have extracted the larger resolved vessel, the existing vasculature, and the smaller vessels are homogenized. Now here is you see, is the tumor, and then the microenvironment, how we modeled this. Here are the data to compare with. These are the diameters of the vessels in color code. These are the pressures in the vessels in color code, and these are the pressure in the interstitial fluid. So now one question is, how do you choose, I mean, where do you stop with the existing vasculature and where you start with the homogenization? There are 2 criteria. One, to choose those with the highest flow, or to choose with the biggest radius. Now, better is the highest flow for 2 reasons. First, the radii, you get them with staining, but they stain also blood vessels which there is no flow, because the vasculature in the tumor is quite disorganized and chaotic, and there are several vessels which are there, but there is no flow because they stop. While with flow, I mean, you get those who are really active, and further, the flow, you can get it with intravital microscopy. So that is the better data. Now I show you how this works. It shows the ratio between the values of the full model and the hybrid model, and you see for the blood pressure it's perfect. In the larger vessel, in the smaller vessel, it's quite good. Then if for the interstitial pressure it's perfect. The flow in the smaller vessel is not so good, but the overall picture is quite good. Okay, so we have that model. Now we have to apply it. We apply it, obviously we are interested in efficiency of drugs. So we have to transport the drugs, and then to evaluate with our model if the tumor still grows or diminishes or dies. So now we go to nanoparticle-mediated transport, drug delivery. For those who are not familiar, okay, first, why nanoparticle? Because the places I in Houston, they're very keen on nanoparticles, and Paolo Di Cuzzi in Genoa also deals with nanoparticles. Obviously, we can also work with chemotherapy. It's just a different concentration, but we are more interested in this for the moment. So the injection, I mean, you start injecting nanoparticle delivery platforms. Delivery platforms, because you can have nanoparticles in nanoparticles. I showed it afterwards. They are injected in the circulatory system. They go around quite fast. I mean, after 5 minutes, they are down in the capillaries. In the capillaries, if you can see, the small yellow dots are the nanoparticles. They segregate out to the wall, where is the endothelium, and interact with the endothelium. They have to stick to endothelium, and then you have to extravasate in the extracellular matrix. Then there they are come in contact with the target cells, and they are absorbed and then endocytosed in the cells, which means that they build a pouch around. There's no hole getting in except for carbon nanotubes, and then go inside of the cell. And there the pH environment is different, so the nanoparticles are dissolved and release the drug. Now that's easy to say, but there are many barriers. First one to the nanoparticles is the immune system. I mean, the phagocytic cells take them away. Then, as I said, the tumor vasculature is quite chaotic. I mean, you have leaky parts, you have non-leaky parts. Then the problem is that the lymphatic system in the tumor is also almost non-existing. That means they have a pressure buildup in the tumor, which is so the fluid flows out, which is against the nanoparticles going in. And then, as I showed at the beginning, that the ECM trafficking, I mean the extracellular matrix is quite chaotic too, which is a barrier to transport. Now, this one you have to solve it with the biodistribution model, which we had done in the past, and all this one you can do it with our model. Just obviously here imaging is very important to see where actually these nanoparticles end up. If you can see, the red dots are the nanoparticles. So these are round particles with two different diameters. You see a large quantity ends up in the liver and in the spleen, where they are eliminated. But however, also important part, it's 10 times less of this, but ends up in the tumor, and where it can release the drugs. So why do they concentrate in the tumor? That is because of the enhanced permeability and retention effect. Here you have the vessel, then I say they are leaky, so there is convection out in the extracellular matrix. However, there already, so the lymph system nanoparticles are eliminated, but those who arrive in the tumor, there is almost no lymphatic flow. There they can concentrate, and it can be at the end quite a substantial number, which is sufficient for therapy. Now I show you we can model this one. This one is a tumor with a small, extracellular matrix with the tumor here, with an existing vessel. We let the tumor grow for 24 days. This one are the isolines for the angiogenic, tumor angiogenic factor. Here is the tumor, here is the necrotic part. And after 24 days, we inject nanoparticles for 100 minutes, then reduce it and stop at 110 minutes. Now let's see what happened. Here you see the neovasculature after 100. I mean, you see the concentration. At the beginning is everything close to the existing vessel, and then it moves inward and exchanges with interstitial fluid. And then from the fluid, it goes to the cells. And you see it here. You can see where our nanoparticles end up. So we are able to simulate where the drugs at the end will end up. Now, come to the last part, which has to do with the use of artificial neural networks, because it's not quite easy to get the parameters for this model. several several parameters you get from experiments, but sometimes it's not easy to extract the values because you have to do many experiments and they are quite time-consuming. For instance, for drug delivery, the killing action, which is in the exchange term, we have to get these from experiments on multicellular spheroids, but the results are not always, they are not always conclusive. So we combine real experiments, numerical simulation, and machine learning to extract these results. To simulate what is going on in the laboratory, we have here the numerical model. And we, on the other hand, in the laboratory have done many experiments. They add this, the drug to the culture medium, and you see with different concentrations. These are the data. You see many, but they are not. And it's not easy to extract conclusive information. You see here, these are the experimental points. You have the radius of the tumor after a different increasing number of days for different concentrations. But it's not sufficient to use to train a neural network. So we use standard interpolation techniques. We use our numerical model and have here the untreated. These are experimental points. We are quite easy to follow that, and that also with 1 nanomole of drug again. So the model works, but if we look with the higher concentration, it's a mess. You can get some information, but you cannot get out a good picture. But neural networks are very good in extracting the overall picture. We use, for the training phase, we give the mass concentration of drug, tumor radius and growth time experiment, and this killing parameter. And then we train it, and then in the recall mode, we just give the same values and get, but different, and get the new value of this killing parameter. Now, you need data for neural networks. So we put here the experimental one. First, we could, with the model, simulate all the intermediate points for the experimental curves. You see, we get all these, we get also all these, and we get also all these already quite more data as we had at the start. But then we can also simulate different experiments, which we did here, to get the full amount of data which are necessary to train our model. And we trained it. Here you see it works quite nicely after 300,000 iterations. The root mean square value is down to 0.018, and the degree of correlation is, I can't see it, but it's 0.99, something like that. And here you see a comparison between the target values, which are these red dots, then the response of the network in the training set, and the blue dots are then for the test set, which we had put aside for testing and not for training. And then with the values we obtained from the neural network, we simulated now the test set. You see we are quite nice, get quite nice results. So that is one application. As far as nanoparticles are concerned, I learned that you use quite a lot at Bernal Institute. Anyway, I will shortly address. These are liposomes, I show at the end. Then also spherical particles. Then we have carbon nanotubes, we have silicon particles, I'll show them shortly here. The silicium particles, they are nice because usually they're quite bigger, larger than 500 nanometers, but they are porous, and here you can put in smaller nanoparticles for imaging, multiple drug delivery, and so you can do all that stuff. Then these are interesting for future, the superparamagnetic iron oxide, because they are excellent for contrast and enhancers in MRI, but they heat up with alternating magnetic frequencies and radio frequencies. And the same is for the gold nanoparticles. Again, they increase the contrast in CT scans, but can be heated with radio frequencies and near-infrared laser. So with this, you can do hydrothermia. I mean, you have again to deliver them, to bring them to the tumor. Then you apply this near-infrared laser, for instance, for gold nanoparticles. You kill the tumor cells, and then they are taken away by the macrophages. Okay, that I have still to model that. So one important thing is that the nanoparticles, to be effective, first they have to adhere to the endothelium to be able to extravasate. And the nice thing with nanoparticles, you can optimize size, shape, surface, and stiffness. And for adhesion, you need also low shear rate, but that is guaranteed by the tortuous nature of the neovasculature. And here we have in general that did some tests with the flow chamber to optimize the radius of the spherical nanoparticles for different shear rates, and we used again artificial neural network. This one is has 2 layers. The other one had 3 layers. And here you see the experimental data you get. Again, here it's difficult to get out a picture. I mean, these are mean values. However, once you have trained the artificial neural network, we are able to predict the relationship between diameter and number of particle density which adheres to the endothelium for different values of the parameters. And here we can get the relationship for shear rates which are not tested, and again for the diameter. So get out now quite a more complete picture. For instance, and it's just the training of the neural network, and we get out with this one the optimal diameter for different shear rates. Okay, that's what we wanted. So I come to the last few slides, but that is the work we are doing now. I mean, they are doing in Genoa, we are looking at the neural networks. Okay, they synthesize liposomes on a microfluidic system, and we want to predict the hydrodynamic size, polydispersivity. I mean, we train, these are the data, and again, you see that, for instance, here for different concentrations, these are empty pore, so that is just cholesterol. These are, you see, for different values of the ratio between organic and aqueous volume. I'm sorry, just here you see this is a liposome, so that the aqueous volume is here, and the other one is the organic one. So from experiments, we get obviously flow rate, the concentration, then this ratio. We train, but we have some results already, but feed it into a machine learning algorithm. We use supervised learning, and what we want to predict is, for instance, here the size, to get the better pictures out of that, but then the encapsulation, because then we want to encapsulate either a colchicine, which is soluble in water, or these one are prodrugs, which go here, which once in the body are metabolized and become a drug. So we have some results, but it's quite interesting too. So I come to the conclusion that I have shown a versatile 5-phase model for tumor growth and drug delivery. The model has been extensively validated. I've not shown all that. We work on it over 9 years now. Also with respect to hybrid solution adopted for angiogenesis. Then I've addressed nanoparticle-mediated drug delivery We use extensively artificial neural networks to get parameters needed for the simulation, optimization of particle diameters, and also now to get to assist synthesizing of new nanoparticles. And outlook, one major problem is the heterogeneity, and for that, since we have many parameters, we need a sensitivity analysis, which is almost done by a doctoral student, Barbara Wirtel in Munich. And then we will extend it for hypothermia and radiotherapy. So I think I'm done. Thank you for your attention. If somebody, I sent a PDF, there are the papers in the end, if somebody is interested. Thank you.
Okay, thank you, Professor, for the very nice and instructive presentation of your work. Is there any question from the audience? Please feel free to use the chat, either the chat or to open your microphone. If not, I have some question. Can the same, and I presume I know the answer because I know that these come from the applications of these methods and this equation come from a different field and were adopted in the description of tumors. But can these principle be applied to the swelling of clays or other non-porous materials that happens to become porous? So
yes,
in particular,
yeah.
I'm thinking of new class of solvents, zeolites, or what they call now metal organic frameworks and metal organic polymers that are closed and they can open when put under gas pressure to separate gases and volatile aromatic compounds.
Yeah, certainly, because we have written all the balance equations at the beginning are material free. They are just based on basic principles and standard irreversible thermodynamics. And then with the constitutive models, they can be adapted to the different situations. And the model gives also this simplified entropy inequality, which gives a framework within which the constitutive relationship have to be formulated to be thermodynamically consistent, and that the consistency can scale up and down from microscale to macroscale and vice versa. Because most models are only written at macroscale and they impose the thermodynamic statement at macroscale and it doesn't downscale.
Yeah, because by experience, I've noticed that working on these soaping materials, that particle size has a huge effect on the absorption properties of the material, even if molecularly and on the nanoscale the material is homogeneous, the size has a huge effect on whether you observe absorption or not.
Yeah.
So.
Yeah. You get the inspiration always from microscale.
Okay.
Then you have to consistently upscale. That's the important thing.
Okay. Is there any other question from the audience? Well, if not, I think people on a Friday afternoon, they want to go for lunch. And I thank you again for joining us and for a very interesting lecture. And I hope we can invite you here in our institute when the traveling is released and becomes easier. So please join me in a virtual applause to Professor Bernhard Schrefler from the University of Padova. Thank you, Professor.
Yeah, thank you very much.